amphibians, cnidarians, plants, and microbes with commercial potential. Much of her work is focused on the exploration of deepwater marine habitats using manned submersibles and remotely operated vehicles to collect unusual organisms that produce therapeutically useful natural products. Her work is highly collaborative, and she's involved in discovery programs for natural products useful against cancer, drug-resistant bacterial pathogens, and parasitic diseases. She's a fellow of the National Academy of Inventors and the American Society of Pharmacognosy. And so please join me in welcoming Amy and thanking her for her lecture tonight.
Get up here. Okay, so today I'm going to tell you a little bit. It's going to go back in time, and it's going to have all kinds of different things in it, but I'm going to tell you a little bit about our natural products program. And we'll see if I can make all these slides work. And thank you all for coming. We greatly appreciate it. So I want to start by thanking the various groups that have funded the work I'm going to talk about today. And one of the most important groups is the Save Our Seas tag. And so if you have one of those tags, you're helping to support this lecture and our Marine Biomedical Program. And that's administered by the Harbor Branch Oceanographic Institute Foundation. Some of the work is funded by the Bankhead Coley program, which comes from the Florida Health. Some of the work has been funded by the National Centers for Complementary and Integrative Health. A lot of our work has been funded by NOAA, a lot of the expeditions, and so NOAA's got a few little issues going on right now, so please try to support NOAA. They're very important to our program and to many things. Some of the exploration I'm going to talk about came from the Center of Excellence in Biomedical and Marine Biotechnology, and then some of the work is funded by the National Science Foundation, which is also having some little issues. But I wanted to start by showing you this map, and this actually comes from our web portal, and you can see up at the top in yellow, you can go out and you can see all about samples that we've been collecting. Our program's been here since about 1984. And so quite a long time, and we've done many, many expeditions. You can drill down on this, I don't know if I can do this for the people who are at a distance. You can click on one of these dots, and then it will come up and it will tell you more information. So if you're interested in the samples after you see this talk, you can go out and look for yourself and explore. You can also then find out information about the taxonomy of the organisms and where they were collected and what depth and lots of different information. You can see pictures of them. If we took a picture while we were in the submersible, you can also see a little video, and I think I have to manually do this. So this is just showing what it looked like in real life, and so you can go see for many, many of the samples, you can actually go watch a little video of that. So I'm going to let it finish here. It's kind of a long video. I have lots of long videos today. I hope you guys have a lot of time. Yeah, and there they're going to collect it. This is a glass sponge, and this is actually, I think, somewhere off Fort Pierce. So there's some reefs out there that John Reed has worked on. So go to my next slide. So I think you know that Florida is an ocean state. So we have the second longest coastline in the United States. The only one with more is Alaska. And our group has been exploring around Florida for a very long time. We've also, you saw from the first map, gone to many other places. But there have been 41 different expeditions around Florida. We've explored 459 sites, and we have over 6,000 samples that we've collected. The samples are in two different places. So down here, you actually can see what is part of our, we call it the Marine Biomedical and Biotechnology Reference Collection, and we were funded by NSF to put barcodes on everything and keep everything super organized, and also to create this portal. And every sample that we collect, we get a little piece of it, and we put it in a little bottle, and we save it. And so there they are. They're here for as long as anyone wants to study these things. These samples are available to people. We also have a very large freezer, and you'll see these are these milk bins, and they're all filled with organisms. And so those are the ones that my chemistry group uses to do the chemistry that I'm going to talk about today. We've used lots of different tools, and these are just three of them. The Johnson Sea Link submersibles, for many years, they were retired in 2010. the UNCW Mohawk ROV, and then we used one, and I have one video that has this Sonsub Innovator ROV that we used, and it had some unique collection tools that they built just for us, where they used like paint buckets and things to keep everything in. But it worked really well. They're very innovative guys at Sonsub. So I have to click through this because the transition stopped working, but I just wanted to show you quite a few of the types of organisms. And I'm hoping that by the end of seeing these, you will be convinced that off Florida, we have lots of biological diversity. And this is weird sponges and other sponges, another sponge. I'm not going to name them all. New species of sponge. But they're very different and they're very beautiful. And each of these is probably its own species. It's so diverse out there. That's a really strange one. And there's sea fans and there's soft corals, the different sponges. Those. And you can see they all look very different. Close up, isn't it beautiful? It's a very strange hexactinellid sponge. And one that lives in the dirt. One that's hard as a rock. Some beautiful corals. Very strange. That's actually a sponge, not a soft coral. Big ugly sponge. And so these are just a small example. We have those 6,000 from around Florida. So this is just a few. But there's amazing biodiversity out in Florida waters. But what about the natural products? I mean, are they diverse too? Well, first I need to tell you what they are in case you haven't heard me speak before, and you might have been to Esther's lecture just a few weeks ago, or maybe a month or so ago. But to me, something called a natural product, it's a small organic molecule that is made by a plant, an animal, or a microbe, and it's not part of the essential biochemistry of the organism. It's not something required to keep the cells functioning and the organisms alive. But it takes energy for an organism to make one of these compounds. And so we don't always know what they're doing in nature, but we presume that there is a reason that they make them. Now, they could be for defense. So this is a coral reef. I'll try to do it this way for people at home. And you can see there's a beautiful picture by John Reed, our ocean explorer. And this is a sponge and all these soft corals around it. And they're all competing for space. So they may make compounds that allow them to better, keep everybody else away. But they may make compounds that allow them to communicate with the next of their species, which might be on that reef that's way in the back back there. That may be where the next one of their species is. So they use chemicals to communicate. And they use them for larval settlement and a lot of different things, but they make a lot of different natural products. So natural products are considered to be inherently bioactive because they're made for a reason and they probably hit some kind of target within a cell. Now we share biochemistry with these organisms and we have many of the same proteins, and that's been shown by doing genetics. And so we can take advantage of these compounds in human health. So I show some natural products that maybe you might be familiar with. So things like morphine, caffeine, maybe you had some coffee. I always say you needed it before this lecture, but anyways, maybe you took some. And maybe you put some Truvia in your coffee, and that comes from the stevia plant. So that's one of the compounds that gives that super sweet to the sweetener called Truvia. This one you probably haven't heard of, salicylic acid, but if you stick this little group on it, acetate, becomes aspirin, and I'll bet there's at least one person in this room that takes aspirin on a regular basis. And almost all the antibiotics come from nature, about 76% of them. And so overall, there's a report that's been published by somebody in the society that I'm in, the American Society of Pharmacognosy, and they say that about 48% of the drugs that are currently used have their origins in nature. They're either the compound themselves or the compound served as an inspiration for developing a better drug. There are marine natural products now. I think there's about 17 that are approved. We had a small role in doing the structure of this one, which is approved for soft tissue sarcoma and in some countries for ovarian cancer. This is another one of the early ones, and it's kind of an example of this is only half the real molecule. They found you don't need the whole thing. Just half of it is good enough to give the activity, and it's very active against metastatic breast cancer. The one on the bottom is now what I would call a blockbuster drug. So this thing is over $1 billion a year in sales. This is an analog of a natural product from the first found in a sea hare, but comes from a cyanobacteria called dolastatin 10. And they tie it to a monoclonal antibody. And some of you may remember we were using monoclonal antibodies to treat COVID. They could go in and bind the protein. But what this actually does is if you have a protein on the surface of a cancer cell and you can make a monoclonal antibody, and it's very specific for the cancer cell, it targets this super toxic compound right to the cancer cell, doesn't get anything else, and it works very well. And they're using this a lot in combination with immune therapies right now. And there's about 100 clinical trials of different monoclonal antibodies. So marine natural products have finally really had a blockbuster drug. So our program is multidisciplinary. It's not just chemists or biologists working separately. We all have worked as a team, and you might have, those of you who got here early saw that a lot of the team is sitting in the back, and we have some of our retirees and people who just came back to visit are up in the back. But we have people who kind of specialize in collections and microorganisms. We have a team that works on chemistry and figuring out what the compounds, a team of people who work on doing biological testing. And we have a team of people who are working on trying to find a large supply of the compound because we're not going to keep going back and collecting these at 3,000 feet off Fort Pierce. So we have to find a better way once we find something that's interesting. So this is, I say, the current team, and it's people who are working there except for, well, not Peter, but some of the work... Peter retired, and we haven't convinced him to come back, but maybe I will by doing this. But anyways, Shirley and John also retired. They're part of the collections and taxonomy team. Peter's the microbiologist. Esther Guzman is our cancer biologist and immunologist, comes up with all these incredible assays. The people on the bottom here, Priscilla, Jill, and Jennifer, are on the chemistry team. And Jennifer actually isn't working with us, but she had a baby and needed to live a little bit closer to her home because she was driving all the way back and forth to Palm Bay. So, but she did a lot of the work I'm going to talk about. And Megan Conkling just finished her PhD a short while ago, and she worked with Shirley and is starting a more independent program and works on sponge cell culture, trying to solve that supply issue. And at the end there you see Dee Harmony and Tara Peterson, who are the people who are working in the bioassays right now. So I'm going to show you really briefly the natural products process, so I don't have to tell you too much about it. But you can go to the YouTube there on the bottom or just search Ocean Science Lecture, Amy Wright Natural Products or something like that. And you'll actually can watch all of this in great detail with demos and all that, where I stood up here and tried not to spray stuff on people in the front row, but I'm not going to do that today. First thing we do is we make an extract. So if you made a cup of coffee, you extracted a natural product today. So you're all natural products chemists because if you made that cup of coffee. But it has the caffeine and flavor components. This is showing a thin layer plate, and the thin layer plate shows all these different compounds, each one of these dots. And so when we make an extract of a sponge, we get this big mixture. So the next step we do is we do a type of chromatography. We use something called a combi flash system. And you might be able to see, it's kind of hard to see probably from the back, but there are all these little peaks going up. We often call this a peak library because we collect each of those into separate tubes, and that's what we end up sending for our biological testing. This just are HPLCs to show that instead of looking like this awful thing, it actually looks much more pure, maybe not totally pure. And this is what I said we test for bioactivity. We then go on and do the structure using all kinds of NMR spectroscopy and mass spectrometry. So that's the chemistry process, and I'm going to skip all of this when I get to the end. I'll just show you a flow chart. So one of the things that's kind of new in our lab is that we have all these specimens. There's about 30,000 samples in the freezer. I would guess about half of those are really big enough for us to get much chemistry out of. But how would we want to decide which ones we want to work on? I mean, we don't have the resources unless somebody wants to give us $50 million. Then we could do it. I don't think it's not coming from NSF right now. So 30,000 samples, how do we decide? Well, we use something called metabolomics, and it's a fairly new technique. And what it does is it takes mass spectrometry, and we run our samples through a mass spec machine, and then we get a chemical fingerprint. Some people call this a footprint. I prefer to say fingerprint. And then we can do statistical analysis to look at how similar or dissimilar they are. And I put Priscilla up here because, honestly, she is the person who went and learned how to do all of these methods. And she really likes mass spec, which is a good thing. And she managed to figure out how to do all of these methods and bring it all into our laboratory. And I wouldn't be talking about any of this if she had not done that. So this is a mass spectrometer. Well, how it kind of works, really oversimplified. But we have an HPLC, which then separates the compounds, and then they have to get charged. So we have to put like a hydrogen atom or a sodium atom on them. They get charged. Then they will be accelerated through a zone of acceleration. They go through a flight tube, and they move through that, and then they go through a reflectron, and they come back to our detector. And the detector is able to very carefully detect the difference in the masses of compounds. And the things that are smaller will fly through the whole system faster than the other ones. And so where they land ultimately and how fast can tell us a little bit about that molecular weight. So in this next slide, what we want to do is measure an accurate mass. So our instrument will do that. And the basis of that, I always have to give you guys too much chemistry, is that most of our organic molecules are made of carbon, hydrogen, nitrogen, and oxygen, and they all have multiple isotopes, but they have one that's the major isotope. And so we can take a molecule, and I just used caffeine as an example, and it has this formula C8, I have to look over here, H10N4O2, and we can then calculate using the most abundant isotope what that would be for each of these based upon the number they have. And we end up with this accurate mass number. So our mass spec will measure that. And so if we see that, we might be able to say, well, okay, that might be caffeine, except I need to go back one step and say it has to be protonated. So what we would really see for caffeine is 195.0802. And our machine will actually measure to about the third decimal place, and that's what a lot of systems do. So getting accurate mass is really nice because then we get our mass spectrum and you can see these accurate masses and you can start to do various of the things, this statistical analysis. So I went through this really fast, but we have this project. It's called the Florida Metabolome Project. We divided up the state and then we took samples from all these different things that we thought were different. And we made extracts, we put them in the mass spec, and we got the mass spectra. And then Priscilla, along with Jennifer Sandel, who helped gather all the data, but Priscilla brought it in and created a database. So when you put things in, and I'm sure you can't read this, but this is all these exact masses of one of our samples. And then you can just click, and it will tell you, well, that might be a compound called zestalide. Or it might be any of these other ones, forbasin B, and then some other very long names. But we can start to figure out what might be in our samples by comparing what's this accurate mass to this database that's been developed. And then you can also do this thing called principal components analysis. And it looks at whether things are similar or different. So is that chemical fingerprint similar or different than others? And we decided to focus on the ones that are different. So you see these are very different from all of these down here, and then some of these are different. And this just shows the fingerprints, and hopefully you can see that they do, in fact, look a little bit different. And in fact, if we blew this up and looked at the actual masses, you would see that they're very, very different. So we wanted to start to look at the chemistry. So I'm going to walk you through some examples of what the chemicals are that we found and some of their biological activities. So this first one is the one I showed earlier, and it has these large masses. And it's an octocoral that John Reed identified as pseudoplexaura as part of our project. And it has these very large masses. And typically for us, the Gorgonian corals usually have much smaller molecular weight compounds. So we were intrigued, like, whoa, these must all be new. And they didn't match anything in the database. So that's all great. So we started to do work. Here's where it comes from. It comes from this area over here called the Florida Middle Grounds, which is a series of kind of these things that come from about 150 feet up to about 80 feet, way out there. And they're very, very exciting to look at. So let's see. Is it not going to go forward? Okay. So let's just run this video. There wasn't high def at the days that we did all these. So there's the organism. And we actually took most of the sound away because there was a guy from BBC in the front of the sub that day talking about weird stuff. We didn't think you wanted to hear it. And Shirley was in the back, and he was interviewing Shirley during the thing for a TV show. Oh. So this is the trick here. I have to make it go. Okay, so now I'm going to talk about more chemistry. I haven't already. But anyway, so we go through this process of doing what we call fractionation, and Jill Roberts did this one. So this is doing the combi flash. And then in this case, normally we would just be looking at biological activity, but in this case, we were actually looking for those target compounds. So we could identify, we call that annotate what the spectra have. So she did that, and we found that there were compounds here, and then more separations were done, and some of this was done by Priscilla and Jennifer. But eventually, we had these various different compounds. And then Jill did all the fun part, which is to use the NMR and to put together what the structures are. And if you want to know more about that, it's in that other video. And we found that this one was a compound called uniolide, and then we got 3 additional compounds. And one of the questions we had was, well, first we didn't expect uniolide because its molecular weight is much smaller than what we saw, but it turns out they dimerize inside the mass spec. Something that we have to keep in mind is that that can happen. But are they bioactive? So I'm going to take a detour now, and I'm going to tell you about a particular assay that has to do with one of the cancer assays from Esther's lab. So autophagy is a process where cells, if they're under low nutrient conditions, they will start recycling things inside those cells. They'll say, well, I don't need this anymore. I'll chew it up, and then I can use it to make a protein I do need, or whatever it is they need. But certain types of tumors that have what we call activating mutations in a protein called RAS, which is an oncogene, can actually hijack the entire process. And they can sit there and do this all the time. But what's worse is they don't just do it to themselves. They can actually affect the cells around them so that they can start to digest the cells around them and then bring those nutrients in, send them through this autophagy process, and get a lot of nutrients to grow. So those tumors can grow really fast because they're able to use this to feed themselves. And some, like pancreatic tumors have this, and they're kind of described as addicted to autophagy. So the idea that we've all had was that if we could shut down this process, maybe we could shut down the growth of these pancreatic tumors. And so down at the bottom is this rather complicated thing. I'm not going to go through all of that. There's a lot of steps to autophagy. But what I wanted to point out is that there's a protein called LC32 that when you start out, there should be very little of it, and then when the process goes on, it grows and grows, and there's more and more and more of it. And then eventually, when the process ends, it should get digested itself because it doesn't need it anymore. So we should see in a normal situation, this goes up and then goes down. But what we see in a lot of ours where we're screening for it is that it just goes up and up and up and up. And we are indeed blocking autophagy. And I hope to show you some of those here soon. So this is the bioassay that Esther's lab set up. And this is a high-content imager, and I think she's spoken about this before, but it's a really fancy microscope that can take images in just seconds, and then you can actually get the data. It'll take like one little well, it'll take 9 images, and then you can actually quantitate how much pink there is, how much green there is, how much blue. And each of these, so blue is the nuclear stain, green is the autophagy, that LC32 protein, and pink is another autophagy protein that's called P62. And so this is the solvent control, and then hopefully what you see is there's a lot more green over here, and where you see kind of yellow, it's the overlap of the fluorescent signal from LC32 and P62. So here it is. This one is actually blocking. Now, we went back and we put the fractions back through the bioassay, and here we saw, wow, great, all of these compounds that we had decided to work on because they look different are actually active. And that was kind of miraculous, to be honest. We weren't sure that would happen, but it did. And I'm going to walk you through some figures here. So this is the assay data for the first one that I've shown you the structure, uniolide. And this is where it's tested at 5 micrograms per mil. And this is where a 1 and 2 dilution, so 2.5, then 1.25, and down, down, down. And then this over here is the methanol control, this final one in the bottom corner. And hopefully you can see that there is, in fact, a lot of green in this. It goes out to here. And there's a value that's measured, which is called the inhibitory concentration 50. So it's where 50% of the response you're looking at, LC32, you have a reduction of 50%. So that value for this one is 1.6 micrograms per mil. And we can also calculate something that's in the micromolar, which allows us to compare directly taking out molecular weight. But just remember, it's around 1.6. So here's the next one. And I hope you can see it's actually, the number there is smaller, and the green goes out further. And the next compound, did I go the right way? Yep, is actually even lower and lower and lower. And it's at 0.18 micrograms per mil. And the final one is really active, really active. It's way down into here. And the value is 0.068. Which turns out to be 180 nanomolar, which is, nanomolar means it's 10 to the minus 9th. So it's really, really potent. And so right now, Esther's lab is working on these compounds and trying to figure out much more about how they work and how they're inhibiting autophagy and what their use might be in the treatment of these cancers that have the activating RAS mutation. So these are in the next step at this point. There's another kind of analysis, statistical analysis, that has kind of been just coming up, and it's called NP Analyst, and it was developed by a lab, the Roger Linnington's lab at Simon Fraser University, and it's kind of a big data thing where you take all the mass spec data for all of the fractions, and you take all of the biological activity for all of the fractions, and then you say, can you tell me which of the ions, that's what you see by the mass spectrometer, is going to be the active one? And so you might see, let's just go to this guy here, this little orange one. So the bigger the circle is, the more active it is. And the darker the color, the better the correlation. And it does, in fact, turn out that the compounds, if we had done this analysis first, it would have told us, go after these compounds that we chose. So it's pretty amazing, the big data stuff, and how it can actually help to predict for us what we should go after. We did it the old-fashioned way, but this might have helped us. So now I'm going to talk about another sponge that's also active in autophagy, and it comes from an area that is called Pulley Ridge. It's out here. And I think I should have, it was, the reason we selected it is, once again, it is way far away, and it looks like it has very different chemistry, and so we said, let's work on that one. And Priscilla put it into her database and found that maybe it was these compounds called netamine D or netamine N, and we had never had those. And so we always like to add new compounds to our library for people to test, because, who knows, maybe those are going to be good for Alzheimer's. We don't really know. So we add to our library all the time. And so we thought it was these compounds, so we said, well, let's prove that it's those compounds. You always have to prove it. But first I'm going to show you this video. Let's see if I can make it work. So Pulley Ridge is kind of this flat area, this part. This is actually called Forsythia land as well, because there's a sponge that we worked on for a long time called Forsythia, and it lives out here. And in fact, this dive, we were trying to find Forsythia, so you'll hear me kind of reference it.
Sample number 1, we're not going to do any photographs. We're at 230 feet. Orange, somewhat lumpy. I think it's lumpier than what we expect. Yeah, and it's growing in with a couple of different types of algae and what also looks like maybe some kind of worm tubes or bryozoan. Anyway, sample number 1.
Okay, so, wait, if I do that twice, there we go. So this is what it looks like once you get it up on deck. I just think it's interesting to look that it was all like swollen up when it's down underwater, and then you bring it up and it's kind of this ugly thing. So it's nice to know what it looks like underwater. And so Priscilla went through the process and started going after those target compounds and did some chromatography and got 2 different compounds, and there's a lot more in here. I actually, just for simplification, I didn't want to put all the different things because there's more compounds as well. But when she did the mass spec of the pure compounds, we had this kind of surprise. So here's what we thought we should see, this 304. This compound would be a netamine. But in fact, the real molecular weight is this 649. So mass spec can be kind of tricky. This is the real molecule, and in the mass spec, it just breaks easily into 2 pieces. Most molecules don't do that, but these ones do. And so really, this is the one that we should have been targeting, and this is the real molecular weight. So we then tested in autophagy and looked to see what the activity would be. And once again, we see some very nice activity. So this is the one where we should see this green again, and so you see all the green and green and green, and way down here, still lots of green. And the value is 0.43 micrograms per mil. So that's pretty spectacular activity. I'm just amazed still. The next one, did it switch? Yeah, goes even lower. So it's almost all the way into here, and so it's at 0.078. So a little bit better than that, the other one from the Pseudoplexaura. So once again, these are compounds that Esther's team is working on to try to determine much more about how useful these will really be. And they're certainly potent, if nothing else. So I'm going to switch over and work on a place called Portales Terrace. And so here's Marathon, and here's Isla Morada, and here's one of my favorite places to go if you ever get a chance to go in a submarine and go somewhere. Choose Alligator Lithohome. Because this place is really super cool. It's just got so many cool things. And we didn't even touch the surface yet. But it's way out there, and I have to start this one for sure from here. And this is just bits and pieces of the whole tape.
Oh, that's kind of interesting over there, too. Two for one at this stop.
And the subpilot is Don Liberatore, who is Shirley Pomponi's wonderful husband.
Yeah,
there's the green light.
One time you get one, and one time you get the other.
It's very hard for me to focus.
Okay, that's sample number 6, and I don't know exactly how to describe that except for to say that it's kind of a creamish yellow in color, and it has kind of volcano or conical shaped lobes with little tiny apical oscules, and it looks like it has kind of a shaggy surface. Anyways, that is sample number and we are at 881 feet. Here's the overlay. For those of you at home, 878 it says here, but whatever. Sure.
Whatever. And so this is one of the unique tools. I don't know how many ROVs have this clam scoop, but it's amazing for picking up sponges really selectively when you're in these areas. Okay, so sometimes we get to play.
Here. Come on. Here, buddy. Here you go. Good boy. Good boy. Look at you.
It's like a dog.
Come here. Come here. Come here. Come here. Come here. Yeah, good boy.
All right.
I won't tell you how long we
As you
sat
say,
there with that
back to
fish,
work.
but anyways, there are lots of these big fish. I think those are called wreck fish, and they're very abundant at these sites that are all along the Florida Keys. There are a
Yeah,
whole
we should
series
probably
of
just
these.
collect one of those, like, the plates like that over there.
Okay.
Just because they're here.
All of these things are
Those.
sponges,
I
sponges,
think
sponges,
it's maybe the
sponges.
same
It's so hard. How do I
shallow.
choose? I need a mass spec out there. I'm telling you. Get that one. Get that one, you know. Don't get that one. We were going into
This will
collect
be
that
sample number 15. It's kind of, I don't know how to describe it. It's got one big fat hollow tube. Looks kind of pinkish in the video, but it's really kind of tan in color. And then it's got this kind of warty, weird base and very smooth surface. That will be sample 15. Sponge like that.
And this particular sample is one that we don't have an activity for yet, but we have a compound that we
This is
have
a
not
collection
yet
of
figured out
samples
its structure,
to-
but we know it's new. So we have a new compound, one or
Looks
more,
like it's hard.
but we haven't yet found an
So
activity
nice
for it.
trying to-
We're still working on it. It's making me dizzy looking at that. Gotta look over there.
There's some live stylaster and some diadema and a reef. Oh my, let's go look at the reef.
The whole top of this is covered with
Section.
corals and sponges, and it's just amazing what's up there. And fish everywhere.
Airless here.
But we don't have any in this video, sorry.
A little oasis.
Oh, look, there's a
It's like a city.
moray.
It's a little town. Oh yeah, there's a moray. That's
Some
a
kind of
moray.
big snapper. Look at that guy. That's dinner.
Let's go mess with the moray.
Okay, so now let's see if I can make it go to the next thing without showing it twice. So once again, this was, it's this guy up here who didn't cluster in with everything. And so we said, well, let's work on that one. And it had a really nice looking mass spectrum. The chromatogram looked great. So Jill took this one on and she did some chromatography, and we ended up with a pretty fast, nearly pure compound, and then got it totally pure. And now I have to detour off to another bioassay. So I won't spend a lot of time on this, but survivin is another target that it's a protein that is found in certain cancers, and the levels of survivin can correlate to prognosis. So no survivin, better prognosis. A lot of survivin, bad prognosis. It keeps cancer cells alive and keeps cells from undergoing what's called apoptosis or programmed cell death, and it's essential for the division of cells. So it helps the cells undergo division, which more cells makes a bigger tumor. It also does all these other things that I won't go into, but all of them, just note, they're all bad for you if you have cancer. You don't want those things to happen. And so the thought that we had was that if we could find compounds that would reduce the levels of survivin, they could maybe be used with chemotherapy, because survivin itself wouldn't just kill the cells, we didn't think, but maybe it would then be another way to work with these really aggressive tumors that have all this survivin. So once again, Esther set up a really wonderful assay, and here this is a different kind of cancer cell. I think this is a DLD1 or A549. She could look at it and tell me. But the pink is the protein survivin, and hopefully you can see that one treated with a natural product from our lab actually has a lot less of that pink than the control does, and it uses this same high-content imager. So pink is what we're looking at there. So this was Jill's one, testing the compounds. We found these two compounds, and one of them was actually active against survivin. It's about 3 micromolar, so it's pretty good against the DLD1 cancer cell lines. And we're still doing some studies on this. And I will tell you, we just found another activity for this, but we're not ready to talk about it yet. It just happened a few weeks ago. So, but it comes from this lovely sponge, which we saw in the video. So this is a little bit of a long story, so I'll try to make it not too bad because I know my time's probably running out. This is a little sea cucumber, a holothurian. And it was collected, as you can see, this is our number. This was collected in 2001. And it came up as what we call bycatch. So it was in a bucket, but we didn't know how it grew. It was just in a bucket, and we thought, well, it looks really cool, so let's save it. We didn't want to throw it away from the dive. And it's been described, this species has been described, but it has always come from where people collected, got it from a trawl or something like that. So nobody really knew how it grew. So I think this is kind of cool because our team gets to go out and even organic chemists can be naturalists and add to the natural history record. So they drew this like most sea cucumbers. Sea cucumbers crawl around in the mud and they eat mud and they poop mud. And that's where they live, in the mud. And so that's where we expected to find this, although it didn't really make a lot of sense, but that's what we thought. So we went, and here's a video, and I just want to point out one thing first. The time is 9:21 in this video, and we usually launch the sub at 8 a.m., so that meant we had spent at least an hour driving around in the mud looking for this little sea cucumber, because we really, really, really wanted it. And so this is actually, well, you'll see what happens, and we got kind of excited. I'll say that. We got a little excited.
Oh, is that there, right out on the top? You see that little white?
I see it. I think that's...
Where the ball is.
Oh, yeah, that's... Oh, wow. Could be.
Could be. I don't know. Okay, which button am I not using right?
Hold on. Let me... I don't want to whack the camera here.
We were both excited.
I think that
I think
might...
that's
yeah,
it. It's
that's
it. Yay!
And there's another one back behind there.
Yeah,
little
all right,
tiny
Amy.
guy.
Good.
Okay, so there he is. Sample number 3, the echinoderm. Woohoo! Woo, woo, woo.
Okay, in the process of picking up a sponge, Eagle Eye Amy spotted one of our target here. So we got one of the
There's another little one right back behind it.
echinoderms. Looks like we got maybe 1 or 2 in this area. We're going to collect them here as well.
We got a search pattern. Woohoo! Woohoo!
Esther was in the back.
Picture. That's him.
And you can see it has these little tentacles, and it filter feeds, and it sits upright. It doesn't crawl in the mud.
So we can just...
And John told me that now that people really know what to look for, they see these on deep coral reefs everywhere, and they're not uncommon at all. They're very common. So it's kind of cool to get to contribute to understanding that. So I'm not going to show the flow chart, but this rather complicated molecule, Priscilla did the purification and got it really pure, and I got to play and do the structure. It was really, really fun looking at all the NMR work. But what I just wanted to point out is that it's actually very potent. It's down in what we would call 51 nanomolar. Again, that's like the 10 to the minus 9th molar at inhibiting survivin and reducing the levels of survivin. And so this compound looks kind of interesting. And it's related to a compound called frondicide, and frondicide actually comes from one of the sea cucumbers that does crawl around in the mud. Oops, hit that. And there's just a small difference here. This is missing in our molecule, and ours has what's called further oxidation in the side chain. But you can buy extracts of frondicide that have frondicide as the active compound. And in places like Japan, people use this as an immune boosting supplement. And apparently there's no toxicity. And the data for frondicide for its anti-tumor activity is really very strong. I've read a lot of papers on it. We haven't done nearly as much work, but we have this nice one from our interesting little echinoderm. Anyways, we love that organism. So I have just one last story that's going to hopefully go pretty fast. We have all these sponges, and they look kind of the same, and they were all identified originally as Jaspis species. So we all thought they were the same genera of sponge, and they all come from different places. So up here in the Panhandle area, and then Middle Grounds, I think that is, and then Pulley Ridge area, and then one down in Tortugas. So they came from all around in the Gulf. And they had activity against methicillin-resistant Staph aureus. So they were active against a drug-resistant bacteria. And so we wanted to find out what are the active compounds. And as part of the metabolome, we also want to know what are the organisms. So here we're going to show collections of 3 of them. Here we go. So there's the first one. And this is from up in that area near the Panhandle. And this one is collected with the Sansub ROV, where the guys were so creative, they emulated our JSL clam scoop and made their own. And they're so good. I mean, they're really smart guys. John went and worked with them and told them what it looked like, and it's all hydraulically operated. I hear some of my group going, and it took forever to collect it. But that's the first sample. We're going to get it eventually here.
Yeah, I see 1, 2, at least a half a dozen in my field of view here. I'm going to zoom in on another specimen while we're collecting the first one. It's definitely the same as this morning. It's a kind of a vase-shaped conical sponge, about 20 centimeters tall, yellow exterior, a large apical oscula, about 5 centimeter hole on top, kind of conical. The top rim and inside is brown and fuzzy. These could be a new species, so we'll want to try to collect it intact.
See that?
So this is in Pulley Ridge area.
So take a look at it.
To the south.
Okay. That thing too? I don't know what that is. Looks like. Okay, we'll get that too.
You hear the enthusiasm, don't you? So the guy in the front was a fish biologist, and John Reed was in the back of the sub telling him what to collect, and he's like, that thing? You're going to collect that thing? I find it just hilarious. But it turned out it was actually a sponge. It looks like a rock to me, but John is very good, right? So let me see if I can go to there. So here are all those samples again, and you can see all the various different ones. And so we actually did some analysis, and this is a different kind. It's called HPLC. But maybe you can see that these 3 on this side, that's why I put them on this side together, all have kind of this big thing. Not much of it in this guy. Maybe I can do it better doing this one. Not much in that one. But they're all very similar in their chemistry. And then these ones, well, they have this other chemical, and it looks very, very different. So, okay, yeah, they're all Jaspis, but they must be different species of Jaspis, if that's right. And remember, the Jaspis is what they do in the field, and they're busy doing all kinds of other stuff, so the taxonomists really don't have much time. Emily Simon looked at this side over here and figured out that the antibacterial compound, the thing that hits methicillin-resistant staph and inhibits its growth, is this compound called iantherin. And then another summer intern, Olivia Hernandez, worked on this series and found that they had samaplen A. And so they have very different chemistry as we think from here, with two different types of compounds. But they're both biologically active against MRSA, all from these. Oh, and then the taxonomists, Chris Diaz and Shirley Pomponi, figured out that these two are a new species of a sponge called Topsentia. This one, I think they're still uncertain. It's a Pacastrella ? And then these are all a new species of Stiletta. And one of the things that we do see is that a lot of these organisms, every time from the metabolome, we're sending so many of these to Chris and Shirley to look at, and they're going, oh, this is new. It's a new species. And so Florida has lots of new species in our deep waters. So if I can go to the next slide, I just have this little diagram that shows these ones in blue have iantherin and these ones have samaplen. And one I'm a chemist, so I like to look at structures. And I just wonder if it's a coincidence that if both of these are dimers, so if you break this bond, you get this molecule. And over here, if you go between these two sulfurs, you get another smaller part. And both of them have MRSA activity. And I don't know, but I wonder if having those dimers has something to do with their antibacterial activity. They also both share this group, which is called a bromophenol. So kind of interesting to people like me. So, chemists are kind of weird. But anyways, so some conclusions for you. I think, I hope that I have maybe convinced you that Florida, this wonderful state, has diverse deep water marine organisms, and that these organisms are rich in natural products. And I've just shown you a few of them. Many of the compounds that we're finding in these organisms are new to science. They've never been seen before. Our team is getting to work out the structures for the first time. And the compounds that we've identified to date, many of them have very, very potent activities that we're seeing in our cancer assays and MRSA. And I didn't talk about some of our other work, which is, in Alzheimer's and others. We have some nice compounds too. I want to say that I think we've really scratched the surface. When we started the metabolome project, I thought, we'll get this done in a year. Honestly, I think it's lifetime's worth of work for the next generation to keep working to get through all of the samples that we have in our collection. But I really want to stress that it's important that if we value these resources as sources of new medicines, we have to be advocates for conservation. We don't want people shrimp trawling through the Oculina Bank, and there are opportunities. John Reed sends things out telling us when to sign petitions to make sure that we don't do that. Those areas by Polly Ridge, there's a lot of sensitive habitat, and hopefully we won't put anything terrible down in the Portales Terrace, but you never know. At one point down off Miami, they wanted to put one of those liquid gas depots or something right on one of the big reefs, and John was able to convince them that was a bad spot. So it's great if we can all be advocates for these reefs. And there's a lot of people. I showed you the current team. There's a lot of people we've worked with, collaborators at other places, students, etc. The subcrew, everyone. I wouldn't be up here without the assistance of all these people. And I want to do a special thanks to Priscilla for all the video editing. She went through a lot of the tapes with me, and then she made them so they were short enough. And her husband, Chip, who's out in the audience, who also helped with that, I think, as well. So I greatly appreciate that. And once again, thanks to all the people who fund us. None of this would happen without them. And with that, thank you for watching. And I think I used up all the time for questions. I tried to do that. But if anybody does have a question, I could try to answer.
I'm just curious, when you're testing activity, is there like a sweet spot you're looking for, or is it just more active the better?
I'm not the world's expert on that, but I've been working with Esther and many other biologists. I think that it depends probably on the assay. So for some things, like when, the one I showed that they're using with the monoclonal antibody, the more active, the better, because they want something super toxic so they can use a tiny, tiny amount. But other assays, maybe you would want, less active is still fine. And it probably depends on each particular assay. And I didn't repeat the question, but I'll repeat the next one. Hi. So the question was, do we need a permit to collect? So the rules are ever changing, probably. But what we actually need is permission to operate the ship and to do the collections from the national fisheries. So it just depends on where you are. But yes, we do have to have some kind of permit. A couple years ago, we went to the Flower Gardens Bank, and we actually needed permits from the sanctuary that we could be there. And overseas, you need import and export permits and all kinds of things. And so that's partly why we shifted back. Plus, our funding was really for working in Florida. So yes, you need permits of some level. But there are also some things you can just kind of collect with a fishing license in shallow water. So the question was that if you are working with like soil microbes, they, to get the compounds to produce, or the organisms to produce them, they need growth factors and other things. And then do our organisms need that? And my guess is yes, they do. But we're actually kind of collecting the whole thing. And in fact, we think that some of the compounds are actually not made by the macroorganism they're inside, but they're by microbes that are present. And we know that whole communities of microbes communicate, right, with each other. And some of these natural products are those communication molecules. And so they interact and can actually elicit the production of natural products. But since we're just kind of collecting the end product, then we don't have to worry about that. Now, Shirley Pomponi's lab and Megan Conkling are growing the sponge cells to get the compounds, and they do a lot of work with growth factors and other things to try to turn on production. And that's a big area of research for them. So the question was, can the compounds be synthesized once they're identified? And the answer is yes. I would say synthetic chemists can take on any challenge, and they may not want to, but they can do it. And so quite a few years ago, I don't even remember what year it was, a long time ago, some of my colleagues can tell me, 1990, was that it? 1990, we took discodermalide with Novartis to the clinic, and they synthesized 100 grams. And it was written up in Chemical and Engineering News as the chemistry accomplishment of the year to make that. But they did it, and they took it to the clinic. So I would say most of them can, but some will be difficult. Maybe AI will do it. Whether So for NOAA, so the question was, have the policies affected our, or at FAUs? I can't speak to FAU overall, but for our grants right now, they're exploration grants, and they're still fine as far as we know. We're hoping we don't suddenly get a message that says, oh, we're cutting you off, because we're in the process of starting to do all the chemistry on the things we collected. But so far, so good. I'm not sure all my program officers that I work with are still there, though. Once again, the established people are still there, and we've communicated with them, but some of the younger people may not be there. So there was another, yeah? So if we get to the point, so when we were doing discodermalide, there was an agreement put in place, a licensing agreement, and then monies did flow back. And so that would be, you know, the next step. So right now we're still way in the early preclinical, right? But if we were able to license it to a company, hopefully they would write a good agreement so that we would in fact get some. And then I think it's shared between the university and the research labs that found it. Yeah. So.
Just by way of comment, one blockbuster drug at the University of Florida, my dad worked as the pharmacology chairman there, brought in millions of dollars to the university.
Yeah, and paclitaxel brought in hundreds of millions of dollars to FSU. So, yeah. So it's just your agreements, and yeah, it should work. So are we, any more questions or?
Perfect. Awesome. Well, everyone join me in thanking Amy. And we'd love to continue the conversation. If you want to join us in the gallery for some cookies, you can chat with Amy. And also, like I mentioned, Sam is outside with our Discovery Pontoon if you want to see a remotely operated vehicle in action. Please join us next week for Dr. Lauren Simonitis talking about inking. Thank you all for coming.